Good morning, my dear students. Welcome to our Zoology online class. Hope you are very happy. Dear students, in the last class we started about one important topic, the replication of DNA molecule. Okay, DNA replication. See, and in the last class we studied about three types of replication process. Actually, this replication is one of the important process takes place during the S phase of the cell cycle. Okay, and during this process, during the replication process, a double-stranded DNA molecule giving two strands, and these two strands are identical or equal to the parent strands. Understand of you? So in the last class we studied about three types of replication process also. Okay, conservative, semi-conservative and dispersive. So these are the three types of replication process we have completed in the last class. Okay, dear students, today also we are going to study about two more important concepts about this replication process. Okay, so listen very carefully, my dear students. See, the first concept of today's class is experimental proof of DNA replication. Experimental proof of DNA replication. Say, two scientists in the year of 1958. Listen very carefully. In the year of 1958, two scientists. One scientist name is Michelson and the second scientist name is Tal. Okay. So these two people togetherly conducted one experiment to distinguish the difference between three types of replication process. Okay, conservative, semi-conservative and dispersive. These are the three major types of replication process. So these two people conducted one experiment to distinguish the difference between these three types of replication process. Okay, so to do this experiment, they used Escherichia coli bacteria. They used Escherichia coli bacteria, and they cultured Escherichia coli bacteria in two culture media. Say, just to imagine, this is one culture medium, this is another one culture medium. Okay, here they cultured some Escherichia coli bacteria. In this culture medium also they cultured Escherichia coli bacteria, and in this culture medium they added some isotope. Okay, they added some isotope. In the first culture medium they added N15 isotope, and the second culture medium they added N14 isotope. Okay, so in both culture medium they took some amount of Escherichia coli bacteria, and in one culture medium they added one isotope. Name of that isotope is called N15, and the second culture medium they added another one isotope N14. Here, the culture medium which is added with this N15 is considered as heavy culture, and the culture medium which is added with the N14, that culture medium is considered as light culture. So this is the heavy culture, this is the light culture. Okay, and they allowed the Escherichia coli bacteria to produce their young ones for many generations. Okay, so many generations continuously the Escherichia coli bacteria is growing or culturing inside both the culture media. Inside the both culture media. Listen very carefully, dear students. Then, at the end of this process, at the end of this experiment, they observe the DNA present in the heavy culture, the DNA present in the bacteria of this heavy culture contains only this N15 isotope, and the DNA present in the bacterial species growing in light culture contains only N14. Okay, N14. So listen very carefully. So this is the N15 is considered as heavy culture and N14 is considered as light culture. Here the bacterial species growing, inside the bacterial species DNA is there. When they absorb the DNA molecule, they absorb the DNA contains only N15. And in here also the bacterial species is growing, inside the bacteria DNA is developed. So inside the developed, then they absorb the DNA of this bacterial species, they absorb the DNA contains N14 isotope. N14 isotope. Understand? Now, with the help of one techniques. Okay, see, the name of the technique is called cesium chloride density gradient identification. So with the help of this technique, they separated both the DNA molecule. The DNA molecule which contains heavy isotope and the DNA molecule which contains light isotope. So these two DNA molecules are kept separately. Okay, kept separately. Understand of you? Now, the DNA which contain this heavy isotope, N15 isotope, is now added into another one culture medium. Okay, so in this culture medium, one substance they get. Okay, the name of the substance is called ammonium chloride, NH4Cl. NH4Cl. So in this culture medium, they get some amount of ammonium chloride. Now this N15 or the heavy top culture or the DNA is now transferred into another one culture medium which contains ammonium chloride. Listen very carefully. See, after 20 minutes, the first replication process is over inside this culture medium. So after 20 minutes, they took a small amount of sample from this culture medium, and they observed the same techniques. Okay, they observed the conditions of the DNA by using the same density gradient identification techniques. They observed the DNA molecules are settled in one band. That band is in intermediate position. Intermediate position meaning position between the heavy isotope and light isotope. Okay. Then after 40 minutes, the second replication process also over inside this culture medium. So after 40 minutes, again they took some amount of samples, and these samples also analyzed by using the same density gradient identification technique. Now they observe that the DNA molecules are settled in two bands. One is in the intermediate position. Okay, one band is in the intermediate position and the second band is in the light band position. Understand? Finally, they concluded that the Watson-Crick dispersive method is the correct method. Okay, understand? Finally, they concluded that the Watson-Crick method of replication, types of replication, is only the correct replication process. Understand? So, dear students, once again repeat this last point. They culture. Both culture medium consist of some amount of Escherichia coli bacteria. In one culture medium, they added heavy isotope N15, and in one culture medium, they added light isotope, that is the N14 isotope. Okay. Then finally, they separated the DNA molecules in both culture medium, and the heavy isotope DNA is now transferred into another one culture medium. That culture medium consists of ammonium chloride. Okay, so ammonium chloride is there. So now this heavy isotope DNA is transferred to the ammonium chloride culture medium. Okay, so they observed the replication process. After 20 minutes, the first replication process was over. So they took some samples and analyzed the samples. When they analyzed these samples, they observed the DNA is settled in one band. That band is in the intermediate position. After 40 minutes, they took some amount of sample from this culture medium, and that samples also analyzed. Now they observed the DNA is settled in two band. Okay, one band is in the intermediate position and another one band is in the light position. Understand? So this is the experiment conducted by Besselson and Stack. Understand? So hope you are understood this topic. So now we completed experimental proof for the DNA replication. Okay. Then now we are going to study about one of the very important 5-mark question: the process of replication. Listen very carefully, dear students. See, actually, in both the prokaryotic organism and also in eukaryotic organism, the replication process takes place with the help of some enzymes. So the first point about this topic is, in prokaryotic organism, there are three enzymes are playing a very important role in the replication process, or three enzymes are needed for the replication process. Name of the three enzymes are DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. Okay, so in prokaryotic organism, DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. These three enzymes are playing a very important role in the replication process. Okay, here this in prokaryotic organism, DNA polymerase 3 playing a very important role in the replication process. Okay, and the DNA polymerase enzyme 1 and DNA polymerase enzyme 2, that two enzymes are very important for the repairing mechanism. Very important for the repairing mechanism. Understand of you? So in prokaryotic organism, three enzymes are needed for the replication process. The enzyme's name is DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. Okay, then in eukaryotic organism, there are 5 enzymes or 5 DNA polymerase enzymes are needed for the replication process. 5 DNA polymerase enzymes are needed for the replication process, and all these 5 DNA polymerase enzymes catalyze the replication process or catalyze the polymerization process. So in eukaryotic organism, 5 DNA polymerase enzymes are there. So all these 5 enzymes are catalyze the polymerization within a short period. Understand of you? Then the last class I told you, in Escherichia coli bacteria, the number of base pairs is 4.6 into 10 raised to 10 base pairs. Okay, totally 4.6 into 10 raised to 6 base pairs are present in Escherichia coli bacteria. Okay, so to complete the replication process in Escherichia coli bacteria, only 38 minutes is enough. Okay, so within 38 minutes, all the replication process is over in Escherichia coli bacteria. Hope you understood this point. Then, next point. When this replication process takes place inside a cell, sometimes some error can occur in the replications. So if any error takes place in the replication process, it leads to mutation. Understand? But the error can be corrected by using some enzymes. Name of the enzyme is called nuclease. So in the replication process, if any error occurs in the replication process, it leads to mutation. But the error can be corrected by using some enzymes like nuclease. Understand of you? Then the next point is, what substance? Deoxynucleotide triphosphate. This substance only giving energy for the replication process. So it's one of the very important one mark question. So deoxynucleotide triphosphate only giving energy to the replication process. Understand, my dear students? Hope you all understood all these points. Then the next point about this replication process mechanism is the replication process takes place in a particular site in a DNA molecule. So in a DNA molecule, the replication process is started in a particular site. Name of that site is called origin of replication. So this is one of the DNA molecule. So in this region only the DNA molecules are start to dividing. Okay, so this region is called origin of replication. And in prokaryotic organisms, okay, in the DNA of prokaryotic organism, only one origin of replication site only there. But in eukaryotic organism, many origin of replication site is there. Okay, so once I repeat this point, in the DNA molecule, the replication process is starting or beginning in one site. Name of that site is called origin of replication. Okay, then next point. Actually, the DNA molecule is a spring-shaped structure. Okay, so the entire DNA molecule cannot be separated for the replication process simultaneously. Okay, so the entire DNA molecule cannot be separated at a time or simultaneously. Understand of you? So the replication process is occur in a small opening. That opening's name is called replication fork. That opening's name is called replication fork, and this replication fork is present in the origin of replication site. Okay, so once again repeat this one. DNA is a very complex molecule, so the entire DNA molecule cannot be separated simultaneously. So the replication is occur in a small opening. Name of that opening is called replication fork. Replication fork. Okay, then next point. See, for the replication process, already I told you it's a spring-shaped structure. DNA is a spring-shaped structure. So for the replication, when the replication process takes place, the DNA strands are separating. The both strands of the DNA molecules are separating. So the unwinding of this DNA molecule is due to one enzyme. Name of that enzyme is called DNA helicase. So the DNA helicase enzyme only separating the both strands in a DNA molecule. In a DNA molecule. Understand a few? Then next point. When the replication process takes place, in one strand, the replication is continuously takes place. That strand is called leading strand. So just imagine this is the leading strand. So in this strand, the replication process takes place continuously. So this strand is called a leading strand. Okay. But in the next strand, opposite strand, the replication process is discontinuous. The replication process is discontinuous. So this strand is called lagging strand. This strand is called lagging strand. Okay, listen very carefully. The replication process takes place continuously in one strand. Name of that strand is called leading strand. And in the opposite strand, the replication process is discontinuous. So that strand is called lagging strand. So the fragments present in the or the pieces present in this lagging strand is called Okazaki fragments. Okay, so here this just imagine here up to here, then here one small gap is there, then again, then small gap is there, then again. Okay, so some gap is present. Discontinuous replication is takes place in one strand, so that strand is called lagging strand. So the fragment present in this lagging strand is called Okazaki fragment. And finally, these fragments are also connected by a enzyme called DNA ligase. Okay, so by enzyme called DNA ligase. Understand a few? Then next point. See, when a new strand is formed in a DNA molecule, the nucleotides, okay, when the replication process continuously takes place, the both strands are moving away. Both strands are moving away. So the nucleotides present in the newly synthesized strands is pairing with the nucleotides present in the parent strand. So here this is a new strand. This is a parent strand. Okay. So here the nucleotides are produced in this new strand. So that nucleotides are pairing or joined with the nucleotides present in the parent strand. And here also one enzyme playing a very important role. Name of that enzyme is called DNA polymerase. Okay, so DNA polymerase only joining these two nucleotides, joining the nucleotides present in both the newly synthesized strand and old strand. Okay, so hope you are understood this last point. Understand? Then the next point, say in the DNA molecule, for the formation of new strand, for the synthesis or production of new strands, one primer is needed. One primer is needed. Maximum a small piece of RNA molecule is used as a primer. Okay, small piece of RNA molecule is used as a primer. So in each and every DNA molecule for the replication process to produce new strand, a primer is needed. A primer is needed. Then the next point. When we add a primer in a DNA molecule, this primer produces a 3-OH end on the ribonucleotides. Okay, when we add a primer, this primer produces a 3-OH end on the ribonucleotides. Then the next point is RNA primer. Okay, now I told you maximum a small piece of the RNA only used as a primer. Then RNA primer is removed. Okay, RNA primer is finally when the replication process is going or moving continuously, and the final step, the RNA primers also are removed from the DNA. Okay, RNA primer also removed from the DNA. Understand? So, when the RNA primers are removed from the DNA, a small gap is present in the newly synthesized fragment. Okay, because we added the RNA primer in the newly synthesized DNA. Already I told you, for the production or for the synthesis of new DNA strands, RNA primer is needed. Okay, so when the final step, the RNA primer is also removed from the strand, newly synthesized strands. So some gap is present in the newly synthesized strand. Okay, then next point. This RNA primer is removed one by one from the 5-end. You all know one end is called 5-end, the next opposite end is called 3-end. So in the newly synthesized strand, the RNA primers are removed from the 5-end. Okay, RNA primers are removed from the 5-end. Then the next point, say, finally all the nucleotides, okay, after removing the primers from the newly synthesized strands, the nucleotides present in the newly synthesized strands is arranged properly. Okay, arranged properly. Okay, arranged in their correct position. And the gap also covered or sealed with DNA ligase. Okay, the gap also, we are seeing how this gap is formed here. Already we have removed the RNA primers, isn't it? So in that area some gap is there. That gap also sealed or covered by DNA ligase inside. DNA ligase inside. So once I repeat this point, the RNA primers are removed one by one from the 5-end. Removed one by one from the 5-end. Then finally all the after removing the RNA primers, all the nucleotides are arranged in a particular position. Now the gap of the newly synthesized strands also covered or sealed with DNA ligase inside. DNA ligase inside. Then next point. As the replication process is continuously takes place. Okay, as the replication process is continuously takes place, two enzymes, helicase and topoisomerase. Okay, helicase and topoisomerase. These two enzymes are pulling the two strands in the opposite direction. Okay, pulling the two strands in the opposite direction, and these two enzymes pulling these strands in the origin of replication site. So here this is the one strand, this is another strand. So in this region only the two enzymes is present. The first enzyme name is called helicase and the second enzyme name is called topoisomerase. So these two enzymes pulling the strand in opposite directions. So it forming a Y-shaped structure. It forming a Y-shaped structure. Understand you? Understand? So once again repeat, as the point of origin of replication, helicase and topoisomerase, these two enzymes pull the strand and forming a Y-shaped structure. Forming a Y-shaped structure. Understand you? Then DNA polymerase, okay, catalyze the addition of nucleotides. Okay, so nucleotides are when the newly synthesized strands are developing in a DNA molecule. Some new nucleotides also formed in the newly synthesized strands. Okay, so here this DNA polymerase enzyme only catalyzes the addition of the new nucleotides in the new strands. Understand of you? Then these nucleotides are arranged in 5 to 3 directions. Arranged in 5 to 3 directions. Arranged in 5 to 3 directions. Understand of you? Then the reason for this condition is it can, okay, the DNA polymerase enzyme can add the nucleotides only in the 3-carbon position. Okay, the DNA polymerase enzyme can add a nucleotide only in the 3-carbon position, so that only the DNA polymerase catalyzes the addition, okay, catalyzes the addition of nucleotides in the new strand in 5 to 3 direction. 5 to 3 directions. Understand a few? So dear students, now we completed the mechanism of replication. So it is one of the very important 5-mark questions. Okay, so listen very carefully. See, maximum 3 enzymes or 3 DNA polymerase enzymes are needed for the replication process in prokaryotic organism. But in eukaryotic organism, 5 DNA polymerase enzymes are needed. So second point is said. Then in Escherichia coli bacteria, totally 4.6 into 10 raised to 6 base pairs are there. So to complete the replication process in Escherichia coli bacteria, only 38 minutes enough. Then the next point, in this replication process, sometimes some errors are happening. These errors leads to mutation, but the errors can be corrected by using some enzymes called nuclease. Then the next point, deoxynucleotide triphosphate is the substance or the molecule. This molecule only giving energy for the replication process. Then in a DNA molecule, the replication process is begin in a particular site. Name of that site is called origin of replication. Okay. In prokaryotic organism, only one origin of replication is there, but in eukaryotic organism, many origin of replication is there. Then the next point, the DNA molecule is a complex molecule, so the entire DNA molecules cannot be separated at a time simultaneously. So the replication process is first occur in a small opening. Name of that opening is called replication fork. Name of that opening is called replication fork. Then the DNA molecule, the separation of strands is done by one enzyme. Name of that enzyme is called DNA helicase. Okay, and in one strand the replication process is continuously takes place. That strand is called a leading strand, and in the opposite next strand the DNA replication process is discontinuous. That strand is called a lagging strand. The fragment present in the lagging strand is called Ukasagi fragment. These fragments are also connected by a enzyme DNA ligase. DNA ligase. Understand of you? When the replication process is continuously takes place, the nucleotides present in the newly synthesized strands is pairing or joining with the nucleotides present in the old strand, and this process is regulated by DNA polymerase enzyme. DNA polymerase enzyme. And for the production of new strand, a primer is needed. Maximum a piece of RNA molecule is used as a primer. Then the next point, primer produce a 3-OH end on the nucleotides, ribonucleotide. Okay. Then the next point, RNA primer. The end of this replication process, RNA primer is removed. So yes, some gaps are present or formed in the newly synthesized strand. Okay. Then next point. These RNA molecules are removed one by one from the fibril. Then finally, after removing all the RNA molecules, the nucleotides are arranged in their correct position, and the gap also sealed or covered with the DNA ligase enzyme. Okay. Then the next point, as the replication process is continuously takes place, at the point of origin of replication, 2 enzymes, helicase and topoisomerase, these 2 enzymes pull the strand and forming a Y-shaped structure. And the last point about this replication, mechanism of replication is DNA polymerase catalyze the addition of nucleotides. Okay, catalyze the addition of nucleotides to the new strand in 5 to 3 direction. Because it can add the nucleotide only in the 3 carbon position. Understand, my dear students? So dear students, now we completed the mechanism of replication and the experimental proof for the replications. Okay, here the mechanism of replication is very important for 5 mark question, and some 1 mark questions also there, then some 2 mark questions also there. Try to complete today itself. We will meet in the next class. Thank you.